Good afternoon, everyone, and thanks for the invitation for John and I to be out with you this afternoon. What we're going to do is cover a bit about one of the latest research initiatives that we've got going at NDSU. And as you maybe guessed in the program, we're titling this the Agribiome Initiative, and I'm going to tell you a little bit more about why we think this is important, and then John's going to cover a bit more about what we intend to do with that program. First off, I want to start with a few introductory comments related to the future of agriculture. And if you think about trends that are driving change within agriculture, it was mentioned this morning, and I think it needs to be emphasized that the consumers that we are working with today, especially younger generations, are much more interested in where their food comes from, and they're interested in understanding production systems, understanding the sorts of things that are used to produce the food that they're consuming. And as a result, we've got to be much more engaged as an agricultural industry in understanding that need and how we continue to talk about and describe the types of production systems that we have. The other thing I think it's important to think about in the context of this initiative is that we need to understand that our agricultural production systems are a very complex ecosystem. The research that's needed to evaluate those production systems is complicated. A multidisciplinary approach is required. And so we need to understand that changing one part of those production systems causes ripple effects or causes changes in other parts of the production system. And I'm glad that this conference focuses more on the integration of some of those things that are critical. And so we get very focused in some cases on production traits or production parameters. In other cases, we get very focused on the economics. But we don't talk enough about the integration also of the social aspects related to our production systems and the communities, the rural sociology that goes with that. And so one of the things that we're particularly interested in with this research is actually the microbial community that is part of these agricultural production systems. And we have, honestly, we've frankly ignored those throughout much of the course of the research that we've conducted thus far. And that's not a criticism of the research that has been done thus far with agricultural production systems. It's an acknowledgement that the tools we've had to study it really haven't existed until just the very recent times. So the agribiome, in our opinion, is kind of what we would describe as the next frontier. And if you think about where agriculture is headed and some of the interest in food production, one of the other change drivers that's happening across agriculture production systems is more regulatory pressure on things like antibiotics, pesticides, herbicides, other inputs. And so we need to have a better understanding of the microbial system, the ecosystem that exists within our agriculture production systems, and we need to work within that ecosystem and understand better how these microbial communities exist and interact within these production systems. And so John's going to share a lot more about some of those aspects of what we want to do and how we intend to do that. What I'll tell you early on here is that the state legislature, through the State Board of Ag Research and Education, really stepped up and helped us enhance our efforts in this area this past biennium. And so the 2019 biennium, the top priority for SBARE, for the Ag Experiment Station, was the Agribiome Initiative. We received $1.16 million in legislative support to begin that process. That's going to allow us to hire a team of scientists and technicians that will work in a couple of key areas. One is focused on the interactions between our livestock production systems and the microbiome. The other is the interactions between our crop production systems and the microbiome. It also provided some additional operating dollars to help enhance and support that research activity. And so I'm going to turn it over to John and let him describe a bit more about the things that we intend to do here. But I'll be back up when John concludes his remarks to take additional questions.
Thanks, Greg. I found this image from an article that was talking about the role of the microbiome and how we should consider our microbiomes in terms of how we consider how each of us has evolved and adapted to the world. So really, as Greg had mentioned, the microbiome has really been kind of, it's been referred to as microbial dark matter, because we knew that it existed, but we really didn't know anything about it because we couldn't measure it, because we didn't have the tools. And now that we have the tools, we've started to explore what microbes are present in us as humans. That's what we've all become familiar with as we take our probiotic supplements and everything else, but also in our livestock and in our plants and in our soils. And as we understand those microbiomes, we can learn more about how to get the best out of our livestock and our crops. So really, we're all composed of far more microbes than there are even our own cells in our bodies. So it really behooves us to understand those microbes and to understand what they're actually doing. And we're really only at the very beginning of that. Understanding how those networks of microbes work together to help us and to help livestock and to help crops to gain nutrients from their environments, and also to protect against disease and to protect against stress. I put this up. This is from a recent publication, just looking at the crop microbiome and or microbiomes associated with plants, and we really have to consider really the microbes in the soil, associated with the root system, associated with the leaves, inside the plants, on the leaves. There are all manners of microbiomes that are actually associated with those plant structures. But the point I wanted to make is that those microbiomes, they don't just assemble by chance. These microbiomes are actually very deliberately assembled, and basically the plant is outsourcing some of its nutrient acquisition and other things to the microbes that are in the microbiome, and it's taking those from the bank that is the soil. So when we've been talking this morning about soil health, that's really what we're talking about, is that there's a bank of microbes in that soil that can actually do the work that is needed by the plant. And the plant actually selects the microbes that it wants by secreting exudates and various other things that actually attract the microbes to the plant. So actually, when we think about the agribiome research, we're actually thinking about not just understanding the microbes and the networks of microbes, but also understanding how those microbes interact with plants, for example. And so it's that crosstalk between the plants and the microbes that's going to be really critical. And we can actually be at a point where we can actually breed plants that are going to select for a more beneficial microbiome. And then once you get to that point of understanding, I think we can make really great strides in agriculture. And again, on the livestock side of things, the expectation is that in the near future, we should be able to breed livestock to select for a particular beneficial microbiome. So we're really looking at the microbes that are in the gut of the livestock as being something that we can select for by breeding, because there is such a strong connection between the microbes and the animals. So as we understand those connections more, we can select for the beneficial relationships, and I guess select against the less beneficial relationships. So really, one of the points I wanted to make here is that this Agribiome research initiative is very much a multidisciplinary initiative. It's looking at the microbes associated with agriculture, but it's looking at that through the eyes of the current livestock and crop research that we're doing, and all of the things that we've been talking about here today are relevant to that. So in building the research capacity, and Greg mentioned that we, as part of this initiative, we're hiring some scientists to work on the Agribiome research area. We actually were able to hire one scientist back in September, and we've been hiring some technicians, and we're currently in the kind of final stages of recruiting two scientists. And I'm going to talk a little bit about those scientists and tell you a little bit of information about the finalists for that position so that you can actually get an idea of the type of scientists that we're looking for this research. So the searches, the two positions that we were searching for is microbial ecology of livestock. You know, that's really just looking at microbiomes associated with livestock. And then this other one we call the phytobiome position, which is looking at the microbes associated with crops. You know, this is a relatively new area of research, but you know, we were pleasantly surprised that there are some really strong candidates out there. We actually put together a search committee from various units in agriculture, and so we got really nice input from very diverse disciplines, from plants and plant sciences, plant pathology, animal sciences, and microbiology. So we've got some really nice input, and I think we've got some really nice finalists. So I just wanted to kind of give you an idea of the type of people that we're looking at for these positions. So for the livestock position, here's one of the people that we're going to be looking at, and this is somebody with expertise in animal science and veterinary microbiology, and their research is you know, looking at host microbe interactions and how those can be manipulated, and that's kind of a common theme for a lot of these people. Again, another person with an animal science PhD and some degrees in animal science, and again looking at this person's looking at immune function and nutrient acquisition and things like that. And then you know, this other person here is again a background in animal science and microbiology, and again person looking at nutrient management. And basically how can we increase the nutrient use efficiency in livestock. And then, so we'll have those people will be coming to campus for interview pretty soon in the next few weeks. For the phytobiome position, again, we have somebody from a USDA lab out in Washington, and this is a person who's looking at interactions of microbes with crops and protection against disease. It's a guy actually from this area. This person is coming out of Colombia and has had a number of research experiences across the US, looking at plant-associated microbes and again plant growth characteristics. And then a person from Manitoba, again, looking at soil microbes and interactions with plants. So really happy that we've got a really nice group of people that are, you know, well qualified for this research, and we're excited to be bringing them to campus in the next few weeks. And you know, if any of you have an opportunity to interact during that time when they're visiting NDSU, we'd be delighted to have your input on these people. So this initiative has just started, but I wanted to tell you a little bit about some of the ongoing research. And you know, I told you we did hire somebody back in September, and we were delighted to get Sam Banerjee. And Sam has wonderful training in soil science and microbiology, and has some great research experience already. That he's bringing to this position, and I just want to tell you a little bit about what he does, and I'm conscious of the fact that it's his research, not mine. So if you have any questions, I probably won't be able to answer them for you, but I will certainly pass those on to Sam. But one of the things that he looks at, and you know, when we look at these microbiomes, as I said, we were only just developing the tools to start to really understand, you know, how these communities of microbes are put together. And one of the things that he's, you know, been working on and he's been at the forefront of discovering is that there are what we call keystone species that are members of these microbial communities, that they have their effect on the community far greater than their abundance would suggest. So if you were to just look at numbers of microbes in the community, these microbes wouldn't stand out because they're not present in high numbers. But when you look at their connection to other microbes, they're very well connected microbes, and that's important for the community structure. So how these communities of microbes are put together. So if you look at on the right hand side, you see these dark spots. Those are Keystone taxa, and that means that they're really critical to how that community is put together. And it's really important for us to understand this because, we want to know how to build good communities of microbes associated with plants and with livestock. So understanding these keystone taxa is important for that. And I did want to point out one piece of data from a study that he did that I thought was interesting and kind of really illustrates the importance of biodiversity, which is something that we've talked about already here this morning. So this is biodiversity at the microbial scale, and also the importance of these keystone taxa that I talked about. So if you look at this is some work that Sam has done prior to coming to NDSU, but it's work he's going to continue here at NDSU, where he looked at different cropping systems. He looked at conventional, no-till, and organic. This is work he did in Switzerland. And if you look at the conventional, no-till, organic, you'll actually see quite a substantial difference between them, and in particular the organic. All of those colored areas, they're basically representing microbial taxa, microbial species. And the diamond shapes are keystone taxa. So you'll see in the conventional system, there are no keystone taxa, so there are really no kind of critical, highly networked microbes in the conventional cropping system. You'll see a few of them in the no-till system, but in the organic system, you see a lot of them. And you look at all those lines represent connections between those microbes. And the ones in the organic system are highly connected. And so there's a lot of keystone species and they're highly connected. So there's a lot of biodiversity in the organic system relative to the other systems. So, that's the kind of thing that we want to try and understand. And that's one of the things that Sam is actually going to be working on. And I just wanted to point out some of the projects that he's kind of started working on. One of the things that we want from the people that are going to be hired to work in this Agribiome initiative, we want them to work with people across the university, across the state. As I said, Agribiome research is multidisciplinary research. And so Sam has already started to work with some people across NDSU and across the state. And here are some of the projects that he's working on, looking at mycorrhizal fungi and salinity, looking at forage yield and nutritive value of alfalfa. Also a project on crop diversity and soil carbon sequestration, and that's out in Williston, REC. And then also working at the central grasslands with Kevin Sedovic and looking at invasive grass species and the role of the microbiome in the invasive grass species. So I'll leave you just by saying again and reiterating that this Agribiome initiative really has huge potential for partnerships because it's really just overlaying another layer of complexity, as Greg had mentioned, and it's just overlaying another layer of complexity on all of the things that we've already been talking about this morning. But it's a really important layer of complexity. And so I think it's really important to have these multidisciplinary research projects. I think it's really important to have projects, particularly when we look at the crop agrobiome research. It's really important to take in the breadth of environmental conditions that we have in the state, the different soil conditions. It's really important to look at the microbiomes associated with different plants under different conditions, so that we fully understand how these microbes work with plants to enhance nutrient use efficiency, to enhance stress tolerance, to create better crops. So with that, I would say if you have any questions, would be delighted to answer them. If there are technical questions on Sam's work, I'll nod and smile and say that I'll pass those questions on. But thank you for your attention.
There is a book out titled 10% Human. I don't know if you are familiar with it or not, but in there they talk about the microbiome in humans, and I'm thinking that the same is true for our cattle. And the second part is, or the main, what I'm trying to ask is the microbiome that's present in plants, which are eaten by the cows, are the two microbiomes interactive?
That's a great question. It's something I really should have pointed out, but it's the connectivity of the livestock with the plants as well. Yeah, absolutely. And I mean, what we eat is going to influence the type of microbiome that we develop, not just in terms of the microbes that we eat, but the types of food that we eat will influence the type of microbes that develop. They're called prebiotics. So, you know, what you eat will kind of select for different types of microbes in the gut. So, yeah, those connections are huge. And then obviously, you know, animal waste that's put back onto the land will in turn impact the microbes that are present in the soil. And you know, we're really just at the beginning of understanding all of that. But yeah, I mean, the kinds of things that we're learning with human microbiome research, it's all applicable to agriculture microbiome research.
I just offer a couple things too. My background is in beef cattle nutrition, and so, you know, in that field, we do study the microbiology of the rumen, and ruminants have an inherent population of microbes in their digestive tract that do fermentation. But monogastrics like humans, poultry, pigs also have a population of microbes that inherit their digestive tract just like we do. Those are all important in terms of nutrient digestion, utilization, and so on. If you think about the newborn calf, for instance, or a newborn lamb, they're essentially in a sterile environment as they are in the uterus. But as soon as they're born, they begin to interact with the environment. And if you watch what their mothers do, their mothers start by licking them. And their mothers have licked them, but they have also been regurgitating and chewing their cud. And so when they lick these newborns, they're actually starting to inoculate those newborns with that microbial population that eventually becomes residents of their digestive tract. And so certainly, as John pointed out, that is a critically important piece of how those systems interact. And so as that newborn calf begins to establish a microbial population in its gut, it's getting some of those microbes from its mother. It's also getting some of those microbes from the environment that it's in through the foods that it consumes or tests or interacts with as it nibbles on grasses, as it eats some of the feed that the mother is being presented with, and so on. And so certainly there's a whole, this is, like I said, kind of the new frontier of research. It's incredibly interesting to think about all those complex interactions that occur, but we are just on the infancy of beginning to understand all of this. Great question.
A question for John. You had that slide up a little bit ago of the three different systems of conventional, no-till, and organic. And my first reaction was, well, that's great. But my second reaction is how can that be? Because what organic systems I have seen depend on as much or even more tillage than a conventional system to function properly. So I was just wondering how they got that result.
I did tell you not to ask me any questions on that because I can't. The research was done in Switzerland, and I'm trying to think, you know, what the. I don't know. I'm sorry, and I'm saying what I said I would say. I don't know enough about what the systems were that there is that difference. So, yeah, I can certainly get you that information, though. I'm sorry.
So my name is Marko Davnic. I'm a professor here. I teach soil sciences. Are you familiar with the Soil Health Institute microbiome project they're starting? Is this going to be something similar to that project? I know they're doing the whole nationwide, but at least the sampling techniques and the assays that you're going to use. Another question is, will this data be available for public and other institutions of higher education Because I understand if NDSU does publish some of this research, it may not be an open source. So let's start with those 2 questions. Thank you.
Yeah, I mean, I'm familiar with that, and you know, we're not part of that, but in terms of data being open to the public, I mean, everything we do is published and is open to the public, and it is freely available. Yeah, all our research findings would be made available, not only in publications but in you know, research reports as well.
You brought up an interesting comment on microbiology within the ruminant of cattle. And the Beef Magazine just had an article this last month on their research that they're doing on improving the micronutrients in the cattle industry to improve its ability to protect itself from certain types of diseases. And for example, you brought up a cow licking a calf. Well, you got to have the colostrum within the calf within 24 hours, otherwise you don't have a calf. And there's been a lot of research done on yearlings and whether or not their rumens are fully developed for feedlots at the time of weaning, or if we should have them on pastures longer. Have you done any research there at NDSU on any of that?
We have done some research related to kind of that weaning transition period. Certainly, you know, at least the anecdotal evidence and some of the experiential evidence indicates that, you know, those calves do transition better if you've exposed them to some of the types of feeds that they would be getting in the feedlot. You know, for any microbial community, if you think about the rumen ecosystem, for instance, any abrupt change to essentially the feed source or the food stocks that they're getting causes disruptions. So you think about our own diet, you know, we're used to consuming certain types of foods. If we switch abruptly to a totally different type of food, we can have some digestive upsets. The cattle are the same way, and so, you know, that's one of the reasons why the recommendations and the way cattle have been managed typically is to recommend that those dietary transitions occur over the course of time, gradually, so that there's not those abrupt changes in the microbial community. The other thing I'd offer in terms of the microbial piece with cattle is there's been over the years a number of products that are marketed, you know, prebiotics, probiotics, that sort of thing, and it's very difficult to see changes in production efficiency measures, growth or feed efficiency and so on, when you're trying to introduce a single organism into literally a ecosystem that consists of millions of different types of organisms. And so we need to get a better understanding on how to utilize those sorts of things in a more cohesive or systematic way as well. Great question.
Thank you. I'm just curious if you're going to research more than cattle, if you're going to include some other species.
Yeah, great question, and John can probably comment on this as well. You know, I haven't reviewed the CVs of the candidates yet. I'm assuming that, you know, the emphasis areas are applicable across multiple species. Certainly, if you look across North Dakota, you know, ruminant animals are our major livestock species: beef cattle, sheep, dairy cattle, and so on. So that's probably going to be where the major emphasis is, but I'll let John comment on that as well.
Yeah, I mean, I think I would agree with that, and some interest in pigs as well from some of the candidates, but certainly the types of research questions they have are really applicable across species, and I think the only limitation is people to work with. So once they have people to work with, I think they have questions that can be asked in a range of livestock species.
Thanks. Kind of extending on that question of what sort of livestock, but for either of these positions or for the current person you have on board, I'm guessing you would have people work on both the microbial system as it impacts potential pathogens, as well as perhaps yield and quality issues as well?
Yeah, so we definitely have on the crop side, we have some people who are very much interested in the pathology side of things. So yeah, that's one of the important aspects of that. Yeah.
One of the things we test for in the soils in a Haney test is the microbiology, the life in the soil. Are you going to be looking at the thing about the Haney test is you can lower your fertilizer inputs depending on the life in your soil then, and the carbon and that kind of thing. So I was just wondering, are you going to do much, or have you done much with how much fertilizer you can actually apply to the soil without it affecting the soil life, or how little you have to, because in theory, if you can get enough fungi and bacteria providing nutrients to that plant, maybe you don't need any fertilizers, or if you do, you know, what is that a avenue you're going down or thinking about, or?
Yeah, I mean, I think that's a great question, and actually it brings up a really good point that this initiative really, I think, builds on and complements the soil health initiative and the work that's being done by people in soil health. It's just another way of looking at what's in the soil that gives you slightly different information that complements the information that you're getting from other sources. Now, we don't have the kinds of answers you're looking for right now, but those are the kinds of things that we hope the microbiome research will bring value to. It's just giving a different type of data on the soil.
We talked about interseeding cover crops in corn earlier. I was wondering is there a way we can find crops to interseed into corn that will select a microbiome that's positive for the corn, and they'll act synergistically?
Yeah, great question, and I don't know that we know the answer to that right now, but certainly as you think about this whole emerging field of the microbiome and the agribiome, I think those are the sorts of things from a systems approach and from a conceptual approach that we would be particularly interested in is what are the sorts of things we can do from a management practice standpoint, perhaps with crop rotation or cover crop interseeding or other management practices that have positive or beneficial effects in terms of enhancing that microbial community that is already present there.
I would just echo that and say that you know, one of the things that's happened with this kind of surge in research in this area is you'll notice that there's a lot of new companies out there that have products that they you know are selling as a microbial product that you can add to your soil or to your crop that's going to enhance its effectiveness. And maybe a lot of those products might end up not being as effective as they'd hope they would be because you're trying to put in a single microbe, as Greg had mentioned, you're trying to put a single microbe into a complex system. You know, maybe the best approach is really in management practices and doing things like you're saying, which is, you know, influencing the microbiome by managing through your management practices and through the type of cropping systems. But really what the microbiome research gives you is that extra level of understanding of what you're actually doing at the soil microbial level.
Anything else for John or Greg? All right, well, let's give them a big hand. Thank you.